What if aging isn't just damage, but misregulation? What if instead of replacing cells, reprogramming, or clearing them, we could reset them back to healthy state without changing what they are? Today's guest is building exactly that future using AI, human data, and RNA therapeutics to identify the master switches of aging inside ourselves. Welcome back to Decoding Longevity. I'm your host, Dr. Raghav Sehgal, joined with my co-host, Dr.
Hi everyone. In our last episode, we explored metabolism and immune aging, and we're seeing the amazing work that is happening at the Buck Institute. And today we're going to move into something incredibly exciting, in my opinion, and it is how can we directly intervene at the gene level of regulation to restore cellular function. So our guest today is Dr. Janine Sengstack, co-founder of Chief Scientist Officer of Genevity, a longevity biotech developing what they call cell reset therapeutics. So her work started during her Ph.D. at UCSF, where she built the foundation of a platform that uses AI and large-scale human data to identify transcription factors, which we're going to explain what it is, that can reverse aging in cells. Janine, it's a pleasure to have you here. Welcome to the podcast.
Very excited to be here. Thanks for having me.
Yeah. So Janine, I'm actually really excited to talk to you specifically to start with about your PhD journey. And partly because it was not just kind of like exploratory science, it was something very translational that sort of became the foundation of your company. And personally, my journey in some ways through my PhD was something very similar. It became what was kind of like these aging biomarkers, which have now become patented. So I really want to sort of do a deep dive for our listeners around how did you really think about your PhD in a way which was really translational, and sort of take us back to the moment, which was what was the core idea for you during your PhD, and what were you really trying to test out, and why do you think it was so high risk and important to translate it?
I'd be happy to. So I did my PhD at UCSF with Dr. Hao Li, who's one of the leaders in aging biology and also computational research and transcription factor biology. And I did a summer rotation with him where I really got to know the lab, which was wonderful, and really dive into aging biology because it's something I'm very passionate about and have been passionate about for a long time, even before my PhD, because aging biology affects everybody. And so it's a really important field to study. And I remember after working with him for a summer and we really liked each other working together, I remember we were standing in line for lunch together one day. And then Howe turns to me and he goes, "Hey, Janine, I have this idea for like a really high risk, high reward PhD project. Do you want to lead it?" And I was like, "Let's go." And this idea that we were so excited about was we were inspired by the Yamanaka factors in terms of in terms of reprogramming capabilities, where you can turn on this collection of transcription factors, which are these regulatory genes, and then turning on just those 4 genes changes a whole bunch of other genes that can take a cell all the way from differentiated back to a stem cell, which is amazing. And that, won a Nobel Prize. And then around the time that I started my PhD, a different lab worked on turning on those genes just a little bit. And they found that if you turn them on a little bit, you can turn cells backwards in time and make them act younger and not go all the way back to stem cell. And we thought that was super exciting. However, it had several risks in terms of challenge for application towards like clinical development, let's say. Overexpressing things is generally a little bit harder to do than repression. And also overexpressing things can have more cancer risk. If you turned on those genes a little bit too much, they turned into a stem cell, which often led to cancer. And so our goal through the PhD project was to see, can we find single transcription factors, so don't even have to do combination. So single transcription factors, and even ideally ones that we can repress to take cells from an old state back to a young state. And so we've developed this computational plus experimental pipeline to test exactly that. And we found actually several transcription factors that were able to do that. And that was around maybe year 5 or so of my PhD. And then we thought, okay, wow, this science is working. How can we move this to patients as fast as possible? And that's when I decided that I wanted to create a company out of that science. And so then I co-founded Genevity. Right at the end of my PhD, we got our first check the week that I graduated, and we've just been
Amazing.
ever since then.
Yeah, no, that is incredible. It's so inspiring, this journey. And I guess like at Genevity, you use the term cell reset therapeutics. So for the listeners, what does that actually mean, and how is it different from approaches like they just mentioned, like the partial reprogramming or even senolytics? And you mentioned that, single transcription factors can turn back the clock. Is that specific to tissues, or is there like a systemic thing that happens? Because that's another problem that we have with partial reprogramming, that it's very localized. So I'm very curious to hear more about how different this is.
Yeah, totally. So our long-term vision or long-term goal is that we could have maybe a once or twice a year drug cocktail that has a specific drug for each tissue that will maybe be like a brain drug, a liver drug, an adipose drug, something like that to bring those tissues back to a healthy state. At Genevity, we think it's pretty unlikely that there's going to be like one drug to rule them all. There
Mm,
are, universal things across tissues that go wrong. That's
Mm.
true. However, there are a lot of tissue specific things.
yeah.
And so our general thinking is an infrequent drug that can bring cells and just kind of nudge them back to health and not to a different differentiation state. I think that's
Mm,
really the reset aspect of our platform. Other, let's say, partial reprogramming approaches are more oriented generally towards pushing something towards a stem cell state or back like that direction. And we want it
Mm.
to just go back to its healthy original state. You can think about it that way because we view that as a very likely to be safe because that is where the
Mm.
tissue should be in the ideal situation. And
yeah.
so, what gets you to tissue specific and infrequent dosing? We think that siRNA, which are silencing RNAs basically, are the way to make this a possibility. And siRNA, that I can talk more about them later, but we're a big fan of Genevity. And thankfully, there's been about 20 years of great science that has been done to really enable this technology. And siRNA have the advantage of very infrequent dosing. You can do like twice a year dosing, for example, and still have repression of your target. And they also have very beautiful tissue specific targeting, which is really important for really high safety.
Yeah.
Mm.
I actually really love this framing that you're putting forward here, which is you're not really thinking about it in the context of replacing cells. You're also not really thinking about sort of erasing their identity, which might be what is happening with reprogramming, right? You're really thinking about sort of restoring them to sort of a healthy
Yeah.
state, which is quite beautiful in a way, right? I really like that. And you quite rightly pointed out, I think one of the really interesting parts here is your approach of sort of using siRNAs. But I will ask you this: siRNAs for a long period of time were considered sort of a little undruggable targets. They were challenging targets. How are you thinking about this at Genevity, and especially with a lot of development that has happened in the siRNA space now? Why go after them in general, and what makes them so powerful in aging?
Yeah. So going after, so specifically in my PhD, I worked a lot on transcription factors and then being able to drug them with siRNA. There's really like the multiple key technologies matured at the perfect time for us to now be
Mm.
able to. 10 years ago, probably would not have been a good idea. Now is the time. And
Mm.
I'll happily explain why that is. So the key things that we care about, right, in creating these drugs, picking the right transcription factor to go after, and then
Mhm.
being able to drug it successfully. Transcription factors are complicated, cool genes that regulate lots of other genes. And so they're really powerful if you can drug them. And in traditional drug development mindsets, is that they've been kind of considered undruggable, right? And however, actually, if you dive into it a little bit more, there's I think 20 blockbuster drugs that target transcription factors.
Mhm.
Mm.
But the nuclear hormone receptor type of transcription factors, which means that they have a binding pocket for a small molecule to go to. The other transcription factors, the like 90 something percent other transcription factors that are not nuclear hormone receptors, were really hard to drug because they didn't have a good binding pocket.
Mhm.
That's where siRNA comes into play because siRNA targets a gene before it becomes a protein at the mRNA level. And so you
Yeah.
don't have to worry about complicated protein structure impeding your ability to drug something. And so the combination of being able to actually drug transcription factors, and then in our case, picking the right one through our reset platform we can then make exciting progress. And then on the siRNA, there's, I believe, 8 FDA approved siRNAs now, and they're very commercially well established. The manufacturing is well established, and the costs are good. So overall, it's like a commodity is, I think, over exaggerated, but it's like a very well established thing now that lots of people are pursuing. And for us, conveniently, some of the key patents are expiring just right for when our drug would be coming.
Yeah, that is so cool. Like before I actually go into my next question, this is totally personal and something that I have been seeing in my research. So I heavily work on DNA methylation, and we also have proteomics. And now with the new technologies, we are able to identify a multitude of different transcription factors that are sort of like working in combination with the DNA methylation. And as we know, DNA methylation is huge in the aging field, and and it works together with those transcription factors. So I'm very, very curious to know that when you actually modifying or working with those transcription factors, have you actually looked at the interaction with the DNA methylation itself, or have you actually looked if the methylation becomes sort of like in a younger profile, like sort of like going back into when you talk about clocks? Have you working on that area?
So that's really exciting that you've been doing that kind of work. The clock work is super cool. That's been working on in the field, not at Genevity, but I touched upon it during my PhD, so I can talk about it from that side. In my PhD work, the model that we worked on was passaged fibroblasts, and we also did an in vivo model as well, but for this conversation, we did passaged fibroblasts, and we turned on or off 4 different transcription factors individually. So it's like ETF3, EZH2, STAT3, and ZFX were the main ones that we focused on. And they led to lots of cellular phenotypes improving, like a little bit more cell division, but not a ton, like a healthy amount more. Proteostasis improvements, mitochondrial improvements. We did check, does the methylation pattern change? And on average, we didn't actually see it changing. There was not a huge undoing methylation from the work that we did in this experiment. And this is just a guess, it's a hypothesis, because we didn't do super in-depth analysis of this, but our hypothesis is that since they were not getting de-differentiated, they were just having some other programs turned on that weren't necessarily methylation related, we didn't see that shift. And so we also didn't see any shift in fibroblast identification genes or fibroblast identity type genes either.
Yeah, no, super interesting. Okay, so moving on to the next part of your work, which is sort of like using this AI and large-scale human data sets to identify those transcription factors or those targets, essentially by mapping which ones lead or drive disease. And that feels like a major shift. So from hypothesis driven to data driven biology, which is quite incredible. So let's go a little bit more into this reshape platform. From what I understand, it integrates the human data, the AI, and the functional biology to identify the targets, and then uses the siRNA therapeutics to silence them. Can you walk us a little bit more in depth on how the pipeline actually works? So once you're silencing those genes, what do you actually see in the experiments? And are you using just the animal models at this stage, or is it just on the cells, or are you actually doing human trials? So let us know, what state are you in?
Yeah. So as a company, we're currently preclinical, but we are gearing up for our first clinical trial. And if things go smoothly, we will be starting that near the end of this year, which is very exciting. And that would be in the type 2 diabetes, with our type 2 diabetes lead asset. So that's an siRNA that in the experiments that we've done in our preclinical models leads to substantial improvements in HbA1c, insulin sensitivity, and has that advantage of very infrequent dosing. So how did we get to this exciting gene? That's through the Reset platform, which I can tell you about. So what started during my PhD work, and then now what we've expanded and grown dramatically at Genevity, is this approach of we can think about a disease as a state of gene expression. That's kind of how we think about it, where you can imagine there's all of our thousands of genes have some level of expression, and versus the healthy state, which has a different level of gene expression. And if you could our hypothesis during my PhD work, and then what we do here, is if you can take that gene expression state from this diseased place and shift it back to a healthy state, then that could lead to cellular benefits and phenotypic benefits, tissue benefits.
Mhm.
And so what we do is we look at large-scale human transcriptomics and genomics data, which to analyze diseased versus healthy gene expression states, and then look at what are the overall patterns of differences between them. And importantly, the best thing to repress is not necessarily the most overexpressed gene, because that would be too easy. Biology is never that.
Mm.
Biology is complicated. And so
Mm.
what we're finding is that the really important genes to go after are the ones that are regulating that disease state. The transcription factor comes in because the transcription factor regulates lots of genes. And so if you can identify the core transcription factor driving the disease state, that one could be the key gene that you should go after to repress. And so we do that kind of analysis along with lots of, like GWAS connections and rare disease connections to add, human mutation data to it to really home in on like what is a list of genes that we think in, let's say, the liver in type 2 diabetics is something that we should repress to treat the disease. So very like tissue disease specific. And from there, then we do experiments to really see, test out the hypothesis. And we can then test them in mouse models. And we've done NHP studies as well to really move that forward into development. And then as part of that process, once we say, hey, this gene is really exciting and we think that we should repress it with an siRNA, there's a parallel arm of Genevity that then creates the optimized drug, because the siRNA you use in a mouse is different than the one that I'll give to a person. And so then we need to go the optimized drug. And we've been able to do that quite quickly and efficiently at Genevity, which has been exciting to see us really come together and grow as a team in that.
Yeah. This is really exciting, and it seems like you're really moving at a very, very fast pace with this platform and sort of already in preclinical, thinking of moving to clinical by the end of this year potentially. I do want to ask you a little more about the cell reset platform
Sure.
and how would you think about it being fundamentally different from some of the predominant approaches that are out there, right? So we have, one sort of arena of things, which is senolytics, right? Then we have, of course, sort of a lot of people betting on cellular reprogramming or partial reprogramming, right? And then there's, of course, sort of the whole metabolic drug target of things, right? You have touched upon this as a platform, how this is different. But if everything works out in your perspective, what does success look like? Would that be, are you treating a specific disease as you are thinking of in the context of diabetes, or would that mean sort of, are you modifying aging trajectories itself? Are you looking at sort of treating specific chronic diseases or potentially modifying aging trajectories as a whole altogether?
Yeah. So I think the way that we think about the platform and how it can apply to aging biology is that we want to go after very precise, targeted, and scalable approaches to go, this is a specific disease that requires a specific drug that we can really move through the FDA approval pipeline and
Right.
make big progress in. For example, having type 2 diabetes dramatically lowers your health span and lifespan and can tons of other comorbidities like neuropathies and various other like amputations, blindness. And so to us, the most like immediately actionable way to make a big difference in the longevity space is to go after like specific diseases that are directly related to health span and lifespan. And so for us, we use our platform to pick the indications that we think are a good fit for that, and then go after the ones that have, let's say, tissues that are relatively targetable with siRNA at this time, which our liver, muscle has had great improvements. Brain siRNA are actually making really big strides. There was, I believe, last week, very recently, there was a Denali brain shuttle, I think, for an oligonucleotide that got approval. Very exciting in that space to really make progress, not only in like direct injections to the brain, but also like peripheral work to get uptake. I think there's a lot of excitement in that space for really unlocking different tissues that are directly related to health span and lifespan and then making those targeted therapies.
Yeah.
Hmm. I have a curious question just before we wrap it up. So, in your vision, you talk about having every year two shots of, just restoring the function of the organs and, target specific organs that are sort of like going out of shape or like becoming unhealthy. Do you see a limitation to it, or how long could you potentially use this therapy? Is that something that is just going to keep renewing and, we're just going to keep on living? Like, just want to have a little bit of your idea on your vision. Is it more, just related to health, and then at some stage, like we stop, the organ will simply stop functioning? Or is it, in your opinion, something that is just going to keep perpetuating?
Oh, I don't foresee us living to like 200 years. So probably not that extreme level, but I my ideal vision is that we can hopefully live longer, but ideally shorten the years that aren't healthy as much as
Mm.
possible. And so if these cell reset therapeutics can help keep the brain healthy for longer, keep the liver healthy for longer, things like that, then hopefully that can have like other multi-organ effects too. You can imagine if you're, for example, if your brain is healthier, obviously so much of your life will be better. But even like the liver, it does so much for your relative, your
Mm.
overall health and metabolism, and making a difference there can have lots of long-term effects. So I guess I would be on the camp that I think these therapeutics will probably not let us live forever, but hopefully, hopefully minimize any years that are unhealthy, and hopefully let us, live those, get that longer health span.
Yeah. Uh, this is really exciting, and almost I would say arguably a new direction from cellular reprogramming and senolytics. And it's a very insightful conversation, Janine, that we have had today, and which highlights that aging might not just be something that we can slow down, but actually something that we could actively reset at the cellular level. And I use the word reset, borrowing from Janine's work. Uh, so,
Yeah.
Janine, thank you so much for joining us. And to conclude, I have one final question, which we ask sort of all of our guests. Who do you think we should invite next to our podcast, and why? We always think our guests are probably the best folks to tell us who should come to Decoding Longevity next to promote good science around longevity. So, who, in your opinion, should be a good person, or maybe a bunch of different people who are doing good in the field and should be here promoting science in the field?
Well, first, I just want to say thank you for the lovely questions and discussion. Thoroughly enjoyed myself.
Mm.
I would recommend Ashley Zender from
Mhm.
Fauna Bio. I
Oh,
don't know
yeah.
if you've got her on the show yet or not, but she's
I'm not,
fabulous.
but I've definitely heard her talk. She's amazing. Yeah.
She's great.
Mm.
She's also, outside of science, she's also a horseback riding friend of mine, and we love horses, and we like pet and brush horses and ride horses together. So,
Oh, how cool! Amazing.
You
Yeah,
should.
no,
uh,
how fun. No, that sounds incredible. Well, thank you so much for coming. This was absolutely incredible. And to our listeners, we'll link all of Janine's work into the show notes, Genevity, and other resources on transcription factors targeting and siRNA therapeutics. So you can just have a look. And if you enjoyed this episode, don't forget to follow, share, and leave us a review. Until next time, I'll see you in Decoding Longevity. Thank you.